Welcome to Cerulean Science for Everyone. My name is Roger, I'm a scientist living in Montreal, and at least once a week we try to talk about science that may interest you. When we think about science, we imagine the highest standard of rational ethics and logic. But science is done by humans, imperfect individuals like you and me. Therefore, scientists, like anyone else, have biases, and unless actively tackling them, they impact the quality and the validity of the results. Okay, so let's talk about the missing diversity in human genetic studies. A commentary on the journal Cell in 2019. First, what is genetics? Genetics is the scientific study of genes and heredity, of how certain qualities or traits are passed from parents to the offspring as a result of changes in DNA sequence. And a gene? Well, a gene is a segment of DNA that contains instructions for building one or more molecules that help the body to work. To link a disease with a specific piece of information in our DNA, researchers use genome-wide association studies, an approach using genetics research to associate specific genetic variations with a particular disease. This method involves scanning the genomes from many different people and looking for genetic markers that can be used to predict the presence of a disease. What is the problem? Well, the majority of studies of genetic association with disease have been performed in Europeans. This European bias has important implications for risk prediction of disease across global populations. Data from 2018 show that close to 80% of the individual data in the genome-wide association studies catalog is from European ancestry. And why is this a problem? Because a disease can be caused by different mutations in different populations despite having a similar outcome. An example is cystic fibrosis, a genetic disease common in Europe and rare in African Americans. In Europeans, the most common causative allele is the F508 in the CFTR gene, accounting for more than 70% of the cases. However, the same allele accounts only for 29% of cystic fibrosis cases in people of the African diaspora. In contrast, another mutation accounts for close to 65% of the patients in South Africans with African ancestry. All this translates in another problem: that different mutations can guide to different treatment choices. Therefore, we need to know which is the cause of the mutation in different genetic backgrounds in order to give the best treatment. In addition, the genetic differences among populations, even if they are small, can affect how efficacious a drug is, or how likely it is to cause adverse events. The take-home message is that this bias on the databases and the studies have significant consequences, such as poor disease prediction and treatment for individuals of underrepresented ancestries. What can we do? We can study diverse populations, increasing our ability to broadly understand genetic disease architectures that will ultimately lead to increased precision in medical care. Thank you for your attention. If you want more information, you can find the link of the commentary in the description of the video. Moreover, if you want to go deeper into science, ethics, and myth representation, I strongly recommend reading the book The Immortal Life of Henrietta Lack by Rebecca Skloot.